Hello, biology students. Mrs. Smith here. I am not feeling very well and I'm going home sick, but I'm making this quick video for you guys so we don't fall behind on our schedule. So please follow along with the video. You should have this worksheet in front of you. Fill out the information and they should help you work on some monohybrid or single trait genetic crosses. If you have any questions, just send me an email tonight. I'll check maybe around 7, 8 o'clock. If not, hopefully I'll be back tomorrow and I'll be able to answer any questions that you have. So, performing monohybrid genetic crosses. Some background information. Punnett squares is something we'll be working with today. Punnett squares are mathematical models used to predict the results of a genetic cross. Reginald Punnett is the person who came up with them. He is an English geneticist, and he developed these squares about 30 years after Mendel reported his studies. Remember we talked about Mendel yesterday. He's the father of genetics, and he came up with 4 different laws that we'll see come into play today. But one we're going to talk about first is the law of segregation. Again, law of segregation states that members of each pair of alleles separate when gametes are formed, so that each gamete receives one allele or the other, but not both. The law of segregation is going to help us explain what we will find in each of the alleles, which will then be what we will find in each of the gametes, which we'll then use to create our Punnett squares. So it says the law of segregation helps make it possible for us to predict what the results of a genetic cross will be. When we use a Punnett square, the gametes provided by the parents will contain only one gene from each pair. For example, if a father's genotype for a particular characteristic is big F, little f, during meiosis, the 2 alleles will separate. Half of the gametes will receive the big F, half will receive the little f. So we can see this little picture of meiosis. This should look familiar. We know that during meiosis, the first thing that happens is the chromosomes will duplicate themselves, so we could represent that here. We know during meiosis 1, the homologous pairs separate, so we can see here and here. And then we know during meiosis 2, the sister chromatids separate. So if you look at any of these resulting sperm, you can see they have either the allele for the big F or the allele for the little f, the recessive trait, but neither sperm has both alleles. That's what the law of segregation is trying to tell us. Last thing right here, we might see some of these symbols. P stands there for the parental generation. F1 is the first generation from that cross. And then sometimes you will see that animals are then bred together, and that's going to create your F2 or your second generation. So using that information, let's go through some Mendelian inheritance examples. This means that our first set of examples all abide by Mendel's 4 conclusions that we talked about yesterday. In particular, the fact of the law of segregation, which is actually always going to apply unless there's disorders, but also the dominant and recessive. So there's going to be one dominant allele, one recessive allele, no mix in between or anything like that. All right. Our first example, in guinea pigs, black hair is dominant over white hair. So let's assign some alleles. Black hair will pick a big B. Usually we just pick the first letter of the dominant, and white hair is going to get a little b. So note we're not giving it a little w. We want to stick with the same letter. Cross a male heterozygous black guinea pig. We know heterozygous means one of each. So that male guinea pig will have the genotype big B, little b. And we're crossing that with a female white guinea pig. Well, it told us that white hair is recessive. In order to show the recessive trait, we need to have 2 recessive alleles. So the female guinea pig is little b, little b. What we know now is that we need to separate those alleles. Just like what we talked about above, the big B and the little b during meiosis are going to separate so that each gamete will get either the big B or the little b, but not both. If we look at the white guinea pig, her eggs are going to get a little b or a little b, but not 2 little b's. Next thing we have to do is fill in our Punnett square. For each box, you need to look at what allele was written above it and to the left. So we need to carry down our big B and then carry across our little. Carry down the big, carry across the little. Do that all the way through. And we've now filled out our Punnett square. Next thing we need to do is come up with our genotypic ratio. Remember, the genotype is the combination of alleles. So we're going to start with our big B, little b. It's the most dominant one we have on there. And if you look, count the boxes, we have 1, 2 of them. We're making a ratio, so make a colon. And what else do we have? We've got little b, so let's write that on there. And if we look, we have 1, 2 of them. If we look in our square, that's all we have. There's no other options. The last thing we want to do is reduce. A 2 to 2 ratio can be reduced to 1 to 1. So your final answer would look like that for the genotypic ratio. All right, phenotypic ratio. Phenotype is how we physically describe traits. So we need to look back at our genotypic ratio to determine that. Big B, little b. How could we physically describe that? That would be a black guinea pig. And little b, we can describe that as a white furred guinea pig. So that is our phenotypic ratio, 1 black to 1 white. Last question on there is what percent of the offspring will have black fur? Well, if we look, these two are black. So we have 2 out of 4. That is 50%. Let's look at number 2. Oops. Cross a female homozygous black. We know homo means the alleles will be the same, and we're knowing that it's black, which is dominant. So she's going to be big B, and we're crossing that with a male homozygous, so they'll be the same. White we know is recessive, so we're crossing that with a little b. We know the alleles will separate, so that each gamete will get one or the other, but not both. We fill in our Punnett square. And if you notice, everything is the same. Every single offspring is going to be heterozygous for fur color. So our genotypic ratio, we can just write, it's going to be all big B, or we could write 1. And our phenotypic ratio, they will be all black. Or you could have written 1, because that represents all. All right, what percent of the offspring will have white fur? None of them. White fur is little b, and we did not get any of that. So 0%. All right, number 3, it says breed 2 of the F1 generation from problem number 2. So the F1 is your first generation. That's these guys here. So we're crossing 2 heterozygous guinea pigs. So we're crossing big B with big B. We know that according to the law of segregation, those alleles will separate. So the gamete will get either a big B or a little b. And then with the other, a big B or a little b, but not both. We go through and fill in our Punnett square, so we look at what's above and what's to the side. All right, now we need to do our ratios. Genotypic ratio. Start up here. We have a big B, and if you count how many, there's 1. All right, what do we have next? We got our heterozygous ones right there. There are 2 of them. And what haven't we accounted for? Our little b. And we have a 1 to 2 to 1 ratio. There's no way that we could reduce that. That's the lowest form we can get. All right, phenotypic ratio. We want to physically describe these. So big B, what color guinea pig would that be? Hopefully you said black. So we know we've got 1 black. All right, big B, what color would that be? Hopefully you said black. That should have also been black. So we can add on. We had 2 more of those, so we have a total of 3 black guinea pigs so far. All right, little b, so recessive. We know that's going to be white. So our final phenotypic ratio is 3 black guinea pigs to 1 white guinea pig. So that would be the approximate probability of what you'd get in the offspring. All right, those are all normal Mendelian inheritance patterns where one allele is completely dominant and one allele is completely recessive. We're now going to talk about a couple exceptions to that. Our first one is called incomplete dominance. That is a situation in which one allele is not completely dominant over the other, and it results in a blending of phenotypes. So let's look at number 4. Snapdragons are a type of flower that exhibit incomplete dominance. The possible colors include red, which is big R, white, which is big W, and pink, which is RW. So if you see, this is not following your normal inheritance pattern. Normally, a flower should be white or red and nothing in between. But as we can see, if you get the heterozygous condition, you end up with a pink flower. So whenever you have a blending of phenotypes, we're going to call that incomplete dominance. So the problem's asking us to cross 2 pink snapdragons. We just said that pink was RW. So we're crossing an RW with an RW. We know according to the law of segregation, those alleles will separate during meiosis. So an allele or a gamete would get either the R or the W, but not both. Either the R or the W, but not both. We fill in our Punnett square. And then we need to do our ratio. So look and see what's inside the boxes. What does our first generation look like? We can see we got an RR here, and there's only 1 of them. Got some RWs. There's two of those. Oops, two RWs. And we got one WW here. All right, phenotypic ratio. Oops, typo there. Phenotypic ratio. Phenotype, we want to physically describe what these are going to look like. So the big R, if we go back and look in the problem, big R is red. So we're starting off with one red. We have two RWs, which right here tells us that's pink. So we have two pink snapdragons. And WW is our last one. If we look back in the problem, that's white. All right, again, if you're reading through a problem and it's talking about blending of traits, that should cue you in that it's talking about incomplete dominance. All right, another exception to Mendel's rules is codominance. Codominance is a situation in which the phenotypes produced by both alleles are completely expressed. So both phenotypes will fully show through. You're not going to get a blending of traits. All right, number 5, the coat color of some cattle exhibits codominance. For example, the heterozygous condition consisting of one allele for red fur and one allele for white fur results in a roan coat, which is RW, a combination of red and white furs. So in this case, red and white don't mix to form pink. There's no pink cows out there. That would be incomplete dominance. This trait happens to follow codominance, which means the red furs and the white fur are codominant together. They both are exhibited. So if you get one of each, you get red and white furs. So roan is what we're going to call the mixed cows, RW, and we're going to mix that with a white cow, which is WW. All right, law of segregation says that during meiosis they're going to separate. So let's separate our alleles and then work on our Punnett square. All right, genotypic ratio. Let's look and see what we have. We've got two RWs, got two WWs. Now hopefully you notice you can reduce those. So our final answer will be 1 RW to 1 WW. All right, phenotypic ratio. What will RW give us? That's a roan cow, so that's the mix of red and white furs. What does WW give us? That gives us a white cow. So in this cross, we would end up with no completely red cows. All right, next exception is multiple alleles, and it says a gene that has more than two alleles. Common example is blood type, where you can inherit an A, a B, or an O. You'll only get two of them, one from your mom, one from your dad, but there's 3 options out there. And as it says, we'll be tackling these types of problems later. So next week, we're going to spend a couple days on blood typing, doing blood typing examples, and doing a blood typing lab. But for now, just know that is an exception to Mendel's rules, having a gene with more than 2 alleles. Last exception up there is polygenic traits, poly meaning many. So it's a trait controlled by 2 or more genes. Skin color would be a common example of this. In fact, scientists believe it's controlled by more than 4 different gene pairs. It's really difficult to do Punnett squares for these types of inheritance patterns. So it's not something we're going to be doing in this class at all. So don't worry about Punnett squares for polygenic traits, but do realize that it is another exception to Mendel's rules. All right, you should now have a worksheet that looks like this, monohybrid cross practice problems, and now you're going to do these on your own or with a partner so long as you're working well together. Your goal is to finish these by the end of the class period. If not, you definitely need to complete them tonight, but also remember your Chapter 11 workbook is due on Monday. So please make sure you're reading through the problem carefully, filling out your Punnett squares, coming up with good genotypic ratios, reducing when necessary, and coming up with appropriate phenotypic ratios as well. When you get toward the end, there's a couple of review questions in addition to those. Have fun doing your Punnett squares.